Good morning. I wish you all are fine. Today we're going to week 2 lecture, the second quarter of the lecture. And just to emphasize that organic genetics is based on values and means. And when we talk about values, characters are measured and expressed in a metric domain, in which the phenotypic values are actually looking at the individual characters. And in this case, the phenotypic value is genotypic value plus environment, P equals to G plus E. So P is phenotype, G is the sum of all genetic effects, E is the sum of all environmental factors. In this case, the assumptions taken is the mean environmental deviation in the population is 0. In this case, when we take the environmental deviation is 0, the P will equal to G. So therefore, population mean equals to genotypic value, and assume our generation is constant. So as you can see, this is a single locus with two alleles, A1 and A2. We give the genotypic value of one homozygote as plus A, another one as this is plus A, and this is negative A, and then the heterozygote as D. So when D is 0, there is no dominance. D is positive and A1 is dominant over A2. When D is negative, A2 is dominant over A1. When D is positive or negative, A dominance is complete. And when D is more than plus A or minus A, there is overdominance. And the degree of dominance is expressed as D over E. So this is a concept of dominance, codominance, and overdominance. As I said, the pygmy mice is one of the mice that is used in the study to demonstrate the concept of dominance, codominance. This was a work done with the assistance of Professor MacArthur in the 1950s in collaboration with Dr. Falconer, when they wanted to study the inheritance pattern in the mice. Apparently, these mice had a mutation in a gene that caused them to be having a dwarf effect, and the mutation is called PG. So let's see the hypothesis. When they did the experiment, they found out, when they did a breeding experiment, they found out after the cross, the weight of mice at 6 weeks of age is with three different genotypes. Okay, if the mice had a PG, it was 6 grams, but if it doesn't have, it's 14 grams. So in this case, if you look at a mean genotypic value between the two homozygotes, 14 plus 6 divided by 2 is 10 grams. So from here you will know that the value of A is 4 grams, and from the results here you will know the dominance here, the value is 2 grams. That's why you will not see the results as such. Why population mean is important? Because we'll be considering all individuals in the population, and we'll consider all gene frequency in individuals in the population, right? And therefore, when you look at the table, if the population has a genotype of A1A1, A1A2, and A2A2, we now denote the frequency, which is P square, 2PQ, and Q square. As I was saying, that you now put in the value of plus A, D, and minus A. Now what is population mean? Population mean is when you divide P square with positive A, you get P square A. 2PQ, you divide with D, you get such. Q and listen, you get this result. So what is population mean? Population mean is the sum of all this and you get this. Alright? So what does that say? When D is 0, there's no dominance. So M equals A, P minus Q. When D is equal to A, it's complete dominance. M equals to A, 1 minus 2Q squared. If A1 is fixed in the population when P is 1, then the population mean is A. If A2 is fixed in the population Q minus 1, then the population mean is minus A. If the locus show overdominance, then the unfixed population will be outside the range. If several loci, then M, now there's a general equation, M equals to sum A, P minus Q plus 2 sum of D, Q. Alright, so this is one formula that you need to remember. I'll give you some questions for you to work on. Now, what are average effects? Do you know that a population exists and is connected to family structure? Thus, when we deal with transmission of values from parent to offspring, we can't use genotypic value. I'm sure you know why. Because it's not the genotype that is being transmitted, it's the gene that is being transmitted from the parents. So we will get one gene from one parent, another gene from another parent. Alright? So to look at average effect, the definition of a particular gene or allele is the mean deviation from the population mean of the individual which received the gene from one parent and the gene received from the other parent having come at random from the population. So in this case, you can see that as you see that the homozygote is given A1, A2, A2, A2 is negative A, and then this is the values, and you calculate with the frequency. This is the mean value genotypes that is being produced by each of the gene, and then you have to minus off with the population mean, which was given by the earlier slide, and then average effect of the gene will be this. All right, I think you can't really see it, but when you move uh, my video, I think you should be able to see it. Okay, so now very important to understand the additive and dominance. So you know that P equals to G plus E, and within the G there's this additive and dominance and epistasis. The concept of additive and dominance should be understood, and in this case a very simple example of what is an additive gene action and what is a complete dominance. So if the individual has a genotype of big A, it has a positive 4 value, but when it has heterozygote, we see a positive 2. This is actually an additive gene action of A. Whereas in this case, if you see a big B is positive 4, but when it has a heterozygote, also it's positive 4. This is called complete dominance. So this is very difficult when a gene shows complete dominance. You can't distinguish a homozygote big B to the heterozygote. All right. So it's very important for when you do quantitative genetics, if you can find out the genes that is controlled by additive gene actions, because you can do improvement or selection on the expected phenotype. So why is it so important? Because you need to understand the breeding value. And the breeding value is a property of individuals and the population from which the mates are drawn. The breeding value of an individual is the sum of average effects of the gene it carries and sum of the pair alleles at each locus and all the loci. So once you see, you can see that these are the genotypes and what is the breeding value that it carries. So every individual has its breeding value for the breeders to actually choose from. What is dominance relation? It arises from the property of dominance among alleles, the interaction between alleles within the locus. So G plus equals to A plus D. So G is genotypic, A is, we call it additive value, or they call it breeding value, and D is dominance deviation. Now what is interaction deviation? If the genotype refers to more than one occurs, so you have G, GA, GB plus I, there's an interaction between the two genes, and it's an additive combination of these values. If I is not 0, the gene are actually exhibiting epistasis. So for all loci, we will have a question looks like genotype equals to additive plus dominant plus interaction division. Now interaction division is what we call epistasis, which plays roles in quantitative traits. You may have interaction between 2 or 3 more loci. This means simply that the gene action at one locus will not be constant, and we call it the interaction effect. So in this case, epistasis is very difficult for us to quantify in quantitative genetics, and it's nowadays being investigated through methylation effects or epigenetic changes through bisulfite sequencing. So with the new technologies, we're able to understand which are the genes that is showing epistatic effect. All right.